Welcome to MrCScience.com's video on experiment 1.1. This is the first experiment in our biology book. This one you can easily do at home. It's kind of a fun one to do, so you might enjoy doing it. You're going to need your books for this for sure. If you got your book handy, in fact, if you don't, stop at the video, go get your book and come back. Then go ahead and turn to page 24. We're looking for experiment 1.1 using a biological key. Now when you do this experiments, you'll need a sheet of paper or a lab book to write your labs up, because we're going to write your labs up like we would in college. So not quite as intense. We'll take some shortcuts, but the general formatting will be there. And remember, the reason we write up labs is to get used to doing it, so that if you ever become a scientist or do that kind of work, you keep track of your experiment well enough that if you do discover something interesting, you can pass that lab off to your colleagues and they can test it for you and see if they come to the same conclusion. So let's get rolling. Let's grab your lab notebook. And we're going to start top of the page, and we're going to walk through how to write up the lab, and then we're going to do this. This isn't truly a lab, it's more of an activity, but we're going to write it up like a lab. Okay. At the top of your lab, we're going to put our Roman numeral number 1. I'm using a sans serif font, so it doesn't have the little up top and bottom, so it won't look quite as fancy, but it's the general idea. Roman numeral number 1, period, title, colon. Then we're going to list the title of our lab. Title of the lab is using a biological key. Okay, that's our title. Then we're going to go down, we're going to do Roman numeral number 2, period. Oops. This is our purpose. Every lab should have a purpose. The purpose of this lab is to, oops, touchpads are evil sometimes. Experience the use of a I can't spell, key. Okay. After that, we're going to go down. We're going to put in Roman numeral number 3, hypothesis. Remember, a hypothesis is an educated guess. We're going to take a look at our different things. Let's see. A good hypothesis would be, let's see, one of the things we're going to look at is the frog. So a frog belongs to the class, I'm pretty sure a frog's an amphibian. You probably know that as well. Amphibia. We're going to say our hypothesis is a frog belongs to class Amphibia. We could put a period in there. You don't have to. This isn't language arts. Okay. Next one is Roman numeral number 4. Oops. Roman numeral number 4 is our materials. It's what we use in the lab. Generally, this is a very detailed and exhaustive list of exactly what you used in the lab, so that if anyone reproduces your lab, they can use the exact material and hopefully get the exact same results. Ours is pretty difficult. We're going to use a biological key and pen and paper. Okay, those three things. So like I said, this isn't truly an experiment, but we're going to write it up like one just to show. Okay, next one, Roman numeral number 5, period, is procedure. This can be one of the longest parts of the lab. Details in excruciating detail exactly how you carried out the lab, so that it can be reproduced exactly as you did it, thus reducing the chance of experimental error, saying that your hypothesis was incorrect. So for this entire class, our procedure is we're not going to waste your time writing out the procedure. It's almost always going to be as per text. If someone wants to carry this out, they can go find your textbook and read about it. Okay. Then Roman numeral number 6. Where's my V? There it is. Observations. As I say, this is where the magic happens. This is where you observe, you gather your data, you record your data. You do not analyze your data, you save that for another part of the experiment. So what we're going to do in this part is we're going to make a little table. Okay, your table is going to go a little bit further down the page than mine will because I'm just going to walk through the first specimen with you to show you how this works. But create this table, and on the left you're going to write your specimen. The first one we'll do is the butterfly. So we're going to put butterfly under specimen. Okay, now we're going to walk through the classifications here. So what we're going to have to do is you've got your picture of your butterfly on page 25. If you back up in your book to page 21, you'll see you've got a simple biological key. This key is just for this experiment. It is obviously not a complete biological key of every living organism on the planet would be fill a library. It would be huge. Might be possible with computers now, but I don't know that anyone's ever completed one. Generally, you pick your area and you go from there. But let's take a look at this. So on page 21, this is like a choose your own story. So the first thing you're going to do is go to number 1, and you've got two possibilities here. Is a butterfly microscopic or macroscopic, which means visible to the naked eye? Well, you can see butterflies, so obviously they're macroscopic. So if you follow that line across, it says number 3. So that means we go to number 3. Okay, now here's two words you haven't heard before. It's a preview. Is a butterfly autotrophic or heterotrophic? Autotrophic means makes its own food, or maybe we discussed this already. Yeah, I believe we did. Heterotrophic means eats. Does a butterfly make its own food, or does it eat? Well, it eats. So if you follow that line across, it says go to number 5. So go to the next page, number 5. Is the butterfly a decomposer or a consumer? Does a butterfly consume things, eat, or does it decompose things, break down dead material? Well, you're not really familiar with what a butterfly eats. It eats nectar, pollen, or nectar, that kind of thing. So it is a consumer. So if you look, as you follow that across, you'll see it says Kingdom Animalia. So what we're going to do is under classification, we're going to add the letter K in Animalia. K for kingdom, and then Animalia. So once you've added that, then we're going to follow that consumer past kingdom Animalia. So let's go to 6. Does a butterfly have no backbone or a backbone? Well, a butterfly is an insect. An insect has an exoskeleton, so it does not have an internal skeleton. It does not have a backbone. So no backbone says go to number 7. Number 7, organism can be externally divided into equal halves like a pie, but it has no distinguishable right or left sides, or the organism can be divided into right and left sides that are mirror images, or cannot be divided into equal halves. Well, you can't split up a butterfly any way you want to look. You can only go right down the middle between the two wings, and both sides are mirror images of each other. That's called bilateral symmetry. You'll learn later. So the organism can be divided into right and left sides. So number 9. Follow number 9 down. External plates that support and protect, or an external shell or soft shellless body. Now I said that a butterfly has an exoskeleton like an insect. So it actually has, although it doesn't seem very plate-like, you can crush them fairly easily. They have external plates that support and protect. So as you go across, you notice it says phylum Arthropoda. So guess what you do? You add P period for phylum and then Arthropoda. Okay, so we got our phylum Arthropoda. Now if you follow that across past Arthropoda, it says 14. So we go down to 14. Does a butterfly have more than 3 pairs of legs or 3 pairs of walking legs? Well, if you look closely at the picture, you'll see that it has 3 pairs of legs. It is an insect, so as you follow your way across, it says class Insecta. So guess what you do? You add C period for class and Insecta. Okay, continue following that over, and you say it goes to 16. Does a butterfly have wings or no wings? Well, that's obvious. It has wings, so we go to number 17. Okay, 17. All wings transparent, non-transparent wings. Well, butterfly are known for their colorful wings, so not transparent. So we go to 19. Large, sometimes colorful wings, or thick, hard, leathery wings. Well, obviously, large, colorful wings. We follow that across, and at the very end it says order Lepidoptera. So we're going to add that. So order Lepidoptera. Now you notice the order Lepidoptera is where the next number you would go to is. So that means you're done. You've hit the end of the biological key for that organism. So you can go back to the experiment and you can add your next specimen, the chipmunk. As you work your way through, you'll do each of these. Not all of them will have all four: class, phylum, or kingdom, phylum, class, and order. So some will have as few as a kingdom, others somewhere in between. So work your way through. Let's see, a couple problem areas I've run across. Grapes leaves are netted, and bacterium. Bacterium are, remember we talked about prokaryotic and eukaryotic? Bacteria are prokaryotic. They have no um, internal or membrane-bound internal cells. The paramecium, you'll see a bunch of membrane-bound cells in organisms within its cells, so that will be normal. So go ahead and stop the video and work your way all the way through baboon and put them all there. When you're done, go ahead, come back, restart the video, and I'll be here waiting for you. Okay, so you've finished your work. Now I'll let you know, although it's fun to do this anyway, but if you turn to page 34, all the answers to experiment 1 are right there, so you can compare your answers to what you have in the lab. If you got any wrong in your lab, it does not make your lab wrong. I will not mark you off for having it wrong on the lab. It just means that there was some learning going there, or you didn't quite understand from the little picture they got what was going on. So don't worry about that. So okay, so now we go back to our lab, and you've got more than just the butterfly here. You have everything else there as well. I'm going to scroll down here and add Roman numeral number 7 and conclusion. In my class, the conclusion is worth 50% of the lab. So if you forget to do the conclusion, I'll mark your lab off 50%. Now that being said, in biology we really don't do any true experiments. We do exercises and activities, things like that. So I don't know that you will ever have to do your own conclusion in my class. Usually we'll do them together at the end. But if you want to expand or anything, feel free. But you don't have to worry about that. Writing up labs in this class should be an easy A for you. So. Okay, so that going forward, let's talk about conclusions. Whoops. Escape, delete. There we go. Okay, what we do in a conclusion is we restate what we did. So let's say, and we analyze and conclude on our data. So using the biological key, we found that. Now, one of the things we'll generally do is relate to our hypothesis. So our hypothesis was a frog belongs to the class Amphibia. So using the biological key, you should have confirmed that a frog is class Amphibia. So using the biological key, we found that the frog, oops, I forgot this doesn't have word wrap, does belong to the class Amphibia. So our hypothesis was correct. Okay. A lot of times what you'll do in conclusion is analyze your data, take your data, look at it, possibly do any math necessary, look for other observations, comment on those observations, how did they relate or not relate to your hypothesis. But like I said before, biology really doesn't do experiments. We do activities. So our conclusions in this class are going to be very simple and short. So this is your conclusion for the lab. Okay, somewhere on our page here or discussing with you, we'll talk about how to get these labs to me for me to record a grade for you so we can move forward in the class. If you have any questions on the lab or anything related to that, always feel free to email me at mrc@mrcscience.com. Thank you.